Hi everyone, today we're going to be talking about hydrocarbon preservation. So in today's video, we'll be covering how hydrocarbon deposits have become preserved in the past in the rock record and the minerals that they're often associated with, why that is, what kind of reactions form those minerals and why they're associated with hydrocarbons, and then also the microbes that are involved in both hydrocarbon preservation as well as hydrocarbon extraction, and how different types of hydrocarbons can form depending on different microbes and minerals that are involved in their initial deposition and diagenesis. So let's get into it. Before I jump right into today's topic, let me just briefly mention here that I do have a whole series on microbial zonation, or the natural layering of microbial metabolic communities in the environment and sediment. So if after this video you want any more or extra information about the reactions that I'm talking about and the metabolisms that are taking place in the sediment that I will be referencing in this video, then you can check these videos out. But on to today's topic. So basically, organic carbon or fossil fuel preservation in the rock record seems like it should be pretty rare considering how much organic carbon is actually continuously recycled in marine and other aquatic environments without actually making it into the sediment and becoming buried and preserved. Most of it is recycled and just released back to the water column and sometimes even the atmosphere, and then goes back through that cycle over and over again. And we talked about that cycle as well as exactly how much is recycled and exactly how much is eventually buried and preserved in the organic carbon burial and diagenesis video. But long story short, it's a very small percentage that actually becomes preserved in the rock record. And we also alluded to this in the oxic, suboxic, and anoxic video when we talked about all of these different microbial pathways that exist within sediment to break down organic carbon. And so you can imagine that once organic carbon gets past all those barriers, all those different microbial communities, it's very much thinned out compared to what it went into the sediment as, which was already thinned out from what fell through the water column, getting recycled through the water column. So you can see here that very small percentage of organic carbon is ultimately preserved. But once it gets past all of those microbial barriers and communities that are there to break it down, it is preserved in the rock record, and that represents a long-term carbon sink or reservoir for holding carbon for geologically long time scales before some sort of geologic process brings it back up to the surface and exposes it to microbial attack again, or some sort of human activity brings it up to the surface and converts it back to inorganic carbon like CO2 or methane that way. But either way, carbon burial and preservation is a long-term carbon sink. But given the fact that we just mentioned that such a tiny percent of carbon actually becomes buried and preserved compared to the amount that's produced, why do we have so much organic carbon or fossil fuel deposits preserved in the rock record to begin with? Are there certain conditions that favor organic carbon burial over others? Well, it's long been thought that anoxia, or the lack of oxygen, like we talked about in that oxic, suboxic, and anoxic sediments video, promotes organic carbon burial because the lack of oxygen means a lack of oxygen respiring organisms. And without aerobic respiration or oxygen respiration, there are less things to break down or decompose that organic matter, allowing its burial in anoxic conditions. However, more recent findings indicate that the decomposition rates in oxic versus anoxic sediments are pretty similar. And this is because those microbes that we discussed in the previous oxic, suboxic, and anoxic sediments video that live in sub and anoxic conditions are almost just as good, if not better, at breaking down organic carbon as those oxygen respiring or aerobic respiring organisms. In fact, some scientists estimate that they decompose organic carbon even more efficiently than oxygen respiring organisms do because they have lower energy yields in their metabolism compared to oxygen respiration, and therefore they have to take up and oxidize more organic carbon than their aerobic respiring counterparts. But regardless of if they're more efficient or even just as efficient, it kind of takes away that anoxic explanation that we have for large organic carbon or fossil fuel deposits in our rock record. So if we can't explain those by saying, oh, it was under anoxic conditions, then how can we explain those deposits? A more accurate explanation for such deposits is primary productivity. Over here to the right, we see two figures. The top figure shows a normal primary productivity rate, and down at the bottom figure, we see an enhanced productivity rate, an enhanced rate of organic carbon production. That enhanced rate of organic carbon production makes it so that the organic matter is falling through the water column and accumulating at the bottom sediment so quickly that both the oxygen respiring and anaerobic respiring organisms can't keep up with that accumulation rate, and much more organic carbon gets past those microbial barriers than it would had the production rate been slower. In fact, we can see in this graph here that high sedimentation rates increase carbon preservation in both oxic and anoxic conditions. However, we do observe a slightly enhanced organic carbon preservation within those environments that were either sub or anoxic. So it does seem like that enhanced primary productivity rate, as well as the enhanced accumulation rates that it causes, is not working alone. Anoxic conditions seem to help a little bit with enhancing the organic carbon preservation potential. But since we already mentioned that the anaerobic respirers are just as efficient as the aerobic respirers at breaking down organic carbon, why would anoxic conditions enhance organic carbon burial? So there are a few reasons why this might be. One is that much of that organic carbon falling through the water column and depositing at the seafloor is protected by silica or carbonate shells or skeletons. These organic carbon mineral assemblages are disrupted in oxic conditions by macrofauna, and that exposes the organic carbon to further decomposition. However, the lack of macrofauna in anoxic conditions could make the decomposition of these materials pretty difficult given that mineral protection that they have. If we look over here to the figure, we can see in the panel on the very left we have anoxic conditions, and in the panel on the very right we have oxic conditions. In the anoxic conditions in the left panel, we have anaerobic microbial respiration at work. However, in the oxic conditions, we've got macrofauna respiration in the water column and at the sediment-water interface, as well as aerobic microbial respiration in the sediment. And then finally, down below that, not mentioned here, but it is there, is anaerobic respiration. However, these macrofaunal organisms in the oxic conditions that contribute to breaking down those mineral skeletons, mixing up sediment during burrowing and bioturbation, are not present in the anoxic conditions. And this lack of burrowing and bioturbation causing mixing up of the sediment promotes a faster rate of burial of that organic carbon, and faster burial rates means ultimately more organic carbon will become preserved. So now that we've discussed how organic carbon deposits have become preserved in the past, aka periods of increased primary productivity create conditions that favor organic carbon burial, now let's talk a little bit about the types of hydrocarbons or the types of fossil fuel deposits that are made from this organic matter. How do we get different types of fossil fuels? Well, ultimately, once buried, all organic matter is transformed by higher temperatures and pressures during burial into insoluble kerogen. However, kerogen has several different types. Labile kerogens, or more reactive, more easily broken down kerogens that just narrowly escape complete microbial decomposition, ultimately form heavy hydrocarbons when preserved, aka oils. More resistant kerogens, however, those that were a little bit harder for those microbes to break down, and probably more of them were able to get past those microbial barriers and ultimately become preserved, those more resistant kerogens ultimately form light hydrocarbons, aka gases. And lastly, the most refractory kerogens, the most difficult for microbes to break down and the ones that most often get past all those microbial barriers, ultimately form graphite. But during the early diagenesis or early burial history of those hydrocarbons, microbes that are in that environment play a huge role in determining the type of hydrocarbon ultimately preserved. Aerobic respiring organisms modify both the chemical and the isotopic composition of crude oil. Ultimately, the oil becomes more viscous, more dense, and richer in sulfur and nitrogen with increased aerobic microbial modification. And this ultimately leads to the formation of heavy oils, asphalt, and tar. However, anaerobic organisms like SRB or sulfate-reducing organisms that live in the anoxic zone of sediments produce sulfide, and that can heavily contaminate hydrocarbons, making what's called sweet or sour crude. But that's not the only way that these sulfate-reducing bacteria affect hydrocarbon preservation. They also promote the formation of iron sulfides as well as carbonate minerals. And this ultimately reduces the permeability of the reservoir rock. So why is this reduction in permeability important? Well, as we can see in this figure, the permeable reservoir rock, as shown in the center with the water, oil, and gas here, needs to be pretty permeable so we can actually extract what's in it. If the permeability is reduced, then our yields upon extraction become pretty low to the point that they're unprofitable. However, even though microbes may have caused this problem, specific microbes are sometimes employed to fix it. Sometimes these microbes are used to dissolve the associated minerals and increase the permeability or just take it away altogether so that they can get the liquids out. Or sometimes those microbes are used to produce gases which increase the pressure within that area, forcing the oil out. Or sometimes microbes are used to produce EPS. I've talked a lot about EPS in this geobiology playlist of mine on my channel, and it stands for extracellular polymeric substances. Why would microbes excreting a mucousy substance help in this situation? Well, EPS production can help stop undesired injection of water into unconstrained pathways in oil-bearing reservoirs. What does this mean? Well, basically, the microbes produce an EPS plug that redirects water into desirable channels that push the oil out through production wells. And this obviously helps us, and we don't even have to continue supplying those microbes with nutrients after they make the EPS plug, because these microbes can maintain the EPS plug even with very little to no nutrient supply. Microbes are not only employed to help get those fossil fuels out from the ground, but also microbes can help remediate oil spills and pollution by decomposing and dispersing oil films, for example, in which those microbes that can grow like this are typically already in the environment where the oil spill occurred, and all that needs to be done to enhance the remediation or increase the efficiency of this decomposition and dispersion of this material is to supply nutrients like phosphorus and nitrogen compounds for those microbes to do what they do best. But another well-publicized fossil fuel issue is often the contamination of groundwater by benzene. Benzene is a highly toxic and soluble organic compound, making it easily transferable through groundwater into drinking water supplies. However, certain microbes can slow or even fully prevent its dispersion into drinking water supplies. And in fact, we can do this in a variety of different environments because benzene degrading microbes have been identified to be able to use a variety of electron acceptors. That just means that they can use oxygen to respire, or even nitrate, iron sulfate, or carbon dioxide. And so because of their versatility, that allows us to employ these microbes for remediation under a wide range of conditions. Researchers are even attempting to identify the genes expressed during this type of remediation, which, if successful, we could favor the growth of the species that will best accelerate remediation in these circumstances by manipulating the nutrient supply. So we are coming a long way in remediation by making it bioremediation, because microbes are incredible. And if you want to know more about bioremediation and biorecovery, by the way, you can check out my bioremediation recovery video. I'll link it up here in the right corner if you want to check it out. The last thing I want to discuss in this video is the formation of minerals in association with hydrocarbons, and what types of minerals most commonly are associated with hydrocarbons and why that is, how they form, what promotes their formation, the microbes involved in promoting their formation, and all of that. So here I have the title diagenetic mineralization. However, I want to have the disclaimer here that I did discuss diagenesis, especially early types of diagenesis, and the metabolic sequence of degradation of organic matter in my previous two microbial zonation videos. So if you want to know more about those specific topics, you can check out those videos. But we have not discussed the formation of more stable minerals during this diagenetic and burial process. We discussed those that are continuously recycled, like oxyhydroxides and sulfides often, but we didn't really discuss those that are stable and really are often buried and preserved in association with these hydrocarbon deposits. And those include carbonates, phosphates, and amorphous silica. So let's talk a little bit about how these form during diagenesis. So first, let's start with carbonate minerals. Basically, aerobes or aerobic respiring organisms produce CO2, which promotes the dissolution of carbonate. So they're definitely not promoting carbonate precipitation. However, as we go deeper into the sediments, we go from oxic to suboxic to anoxic sediment, and anaerobes or anaerobic respiring organisms that live in the sub and anoxic conditions in the sediment promote carbonate precipitation by increasing pH. In the nitrogen and iron zones, for example, carbonate minerals like rhodochrosite or manganese carbonate, as well as siderite or iron carbonate, may form. However, in the sulfur zone, where SRB or sulfate-reducing bacteria produce sulfide, calcite is formed because iron is tied up in those sulfides and won't be associated in those carbonate minerals. But once in the methane zone below the sulfate zone, again, we get iron carbonates forming once again, aka siderite-like minerals or iron-containing carbonates, and that is because the sulfide is gone at that point. What about phosphate minerals? Well, phosphate is released from organic matter upon organic matter decomposition, and once released into the pore waters, phosphate ions can react with a whole range of metal cations to form insoluble minerals. For example, fluorapatite or a calcium phosphate fluorite mineral can form. However, phosphate is often taken up or adsorbed onto iron, manganese, and aluminum oxyhydroxide minerals. And this pathway is actually not as permanent as insoluble mineral formation. So the phosphate that's actually adsorbed to these minerals is often re-released back to the pore waters because these minerals readily dissolve once they reach reducing conditions, or basically sub to anoxic conditions. So at that point, the phosphate will be re-released back to the pore waters and then maybe react with calcium again at that point and then form these calcium phosphate minerals that get preserved in the rock record, or it will continue to adsorb to other minerals and be recycled in those pore waters till it finally does form an insoluble mineral and get buried and preserved. Lastly, we have silica. Amorphous silica formation occurs during dissolution reprecipitation reactions of previously deposited silica shells and skeletons. So we talked about how a lot of organisms form their skeletons or shells with silica. For example, here we have pictures of diatoms, which are just microalgae that have silica skeletons or capsules. And 95% of these shells or capsules are recycled, and the silica is dissolved and released to the water or pore water as ions. However, the other 5% is buried, where most of it then becomes diagenetically altered. During this diagenetic alteration, they're dissolved, but then reprecipitated as the next most soluble phase of silica. So if you don't know, silica can be a lot of different types of polymorphs or crystal structures. I talk about that in my mineral polymorphism video. If you want to check it out, I'll link it up here to the top right corner. But basically, as dissolution reprecipitation occurs, they continually are reprecipitated as the next most soluble phase. Often this occurs multiple times until they're precipitated as quartz. However, if this doesn't occur too many times, the final mineral phase that does get preserved often retains the outline of the diatom or radiolarian that was dissolved and reprecipitated. And if you want to know more about radiolarians and diatoms and what they are and why they have silica shells and how they form their silica shells via biomineralization, you can see my biomineralization part 4 video all about both magnetite as well as silica biomineralization. But regarding hydrocarbon preservation, associated minerals, and microbial reactions that are involved in such preservation, that is all I have for you guys for this topic, and I hope you guys enjoyed. And if you want to check out the reference I'm using to make these videos, I am using the Introduction to Geomicrobiology by Kurt Konhauser. Wow, that was out of frame. And this is a great book. It's linked in my description below, as well as other minor and supporting references. They're always linked in my description if you want to check those out. And if you want to check out the previous 3 videos I've done over microbial zonation to learn more about the topic that was discussed in this video, you can check out my geobiology playlist by clicking the geobiology playlist rectangle card thing down below on the screen right now. You can click it. There you go. All right. Thank you guys for watching. I'll see you guys next time. Bye.